mechanisms underlying the expression of different transcript isoforms for protein coding and no coding genes in normal and stressful conditions, disease and drug treatments in human and mouse. I would like to invite Professor Glória to give her talk. Thank you very much for your attendance.
Thank you very much, Mariana. Thank you for the invitation. It's a great pleasure to be here. This is my first time in the international meeting on animal venoms. I come from the field of protozoan parasites and nematodium parasites, and after that I migrate to human, but I'm working in collaboration with Dr. Maria Helena. That's why I am now moving to peptides, and I am very happy to be here, not only because everything is new for me, and then it's very interesting. I love to learn new things and see old friends. That's very good. Fernanda was my student during the undergrad, and then it's a pleasure to see her again. And today I'm gonna talk about, let me see here. Well, no, here. Well, yes, thank you. I'm gonna talk about comparative genomics of an important bacteria called Acinetobacter baumannii, and how it responds to a toxin from a spider, Lycosa erythrognatha. Acinetobacter baumannii is a very, very important bacteria for health. Is an anaerobic, gram-negative, opportunistic, non-fermentative, and non-motile coccobacillus. And it is involved in healthcare-associated infections, especially in lung, causing pneumonia, urinary tract, blood, bone, and brain meningitis. It is responsible for approximately 45%, 45% of the isolates of this bacteria are multi-drug resistance, especially to carbapenem. There is a very used antibiotics in clinics, and it is responsible for 20% of infections in intensive care units. Well, there are many ways that a bacteria, and especially Acinetobacter baumannii, can get resistance to antibiotics. It can be a mutation in a gene that has a normal function, and then when it is mutated, the bacteria becomes resistant to the antibiotics. Can be the acquisition of plasmids that come from the environment, from other bacteria by conjugation, or acquisition of transposable elements. And then only plasmids and transposable elements are transferred vertically. And it's an important way to get resistance to antibiotics. And the genes that are most responsible for resistance are beta-lactamases, for beta-lactamers antibiotics, efflux pumps, which are not only important for antibiotics, but for getting different compounds, and then getting the compounds out of the bacteria cells, and then they can be used also for efflux of antibiotics. Porins, there are proteins responsible for changing elements with the environments, or penicillin binding proteins, or PBPs. And then here we are looking at general ways to get resistance. I think the most important is the efflux pump, or change in the target by mutation or modification of the site where the drug binds, or bypass the target, creating a new protein that can do the same function as the inhibited one, or inactivation of the antibiotic. And then this Acinetobacter baumannii is very, very resistant to different kinds of antibiotics. And then we are also searching for new treatments with new antimicrobials. Well, Maria Helena and her group has been working for a long time with toxins from scorpions, spiders, especially the one from Lycosa erythrognata, Venom, and she tested the one called LyeTx I peptide. And it has antimicrobial activity. They showed that this peptide destabilize or disrupt the plasma membrane. That's depend on the concentration. And has antimicrobial activity against pathogenic bacteria such as Escherichia coli, Staphylococcus aureus, and also fungi, Candida krusei and Cryptococcus neoformans. And interestingly, it has low hemolytic activity. And now they resolve the structure by NMR, and it's a very short peptide with an alpha helix like this, and it has many residues that are basic residues, and also many different hydrophobic residues like the tryptophan, isoleucine, leucine, phenylalanine, leucine, and then it's composed mainly by positive amino acids and also this these amino acids that are no polar. And then this peptide is 25 amino acids, the LyeTx I, and it's a little bit difficult to be synthesized, and then they decided to remove some amino acids from the carboxy terminus of this peptide, creating a new one that is minimized, the LyeTx I minimized, but with a leucine in the middle that was introduced during synthesis. And then they are very, very similar, but with introduction of this leucine and reduction in the C-terminus of the peptide, creating the LyeTx I Mn delta K, is 16 amino acids. And then they checked for the activities of this minimized peptide, and why it's so important sometimes reduce the peptide, because a smaller sequence facilitates a rapid production of the antimicrobial peptide, decrease the synthesis cost, increase stability, and accelerate the translational clinical applications. And then in this work, we are testing the reduced version of the LyeTx I. Well, in this paper in 2021, William Lima analyzed many aspects of this new peptide, the reduced form, the LyeTx I, against many different resistant Acinetobacter baumannii that are resistant to carbapenem, and also he compared with a susceptible Acinetobacter baumannii. And then they investigated several aspects. They saw the spectrum, the antibacterial spectrum of this peptide against a collection of carbapenem resistant, they called CRAB, of Acinetobacter baumannii, and also they calculated the MIC and the bactericidal concentration to kill bacteria, the not only the resistant, but also the sensitive ones. They evaluated the activity of this peptide to reduce the biofilm and then the anti-biofilm activity that is very similar to the one, the big one. They evaluated the ability of this minimized peptide to induce bacterial resistance after serial passages with the presence of the peptide, and they saw no resistance at all after many days in the passage with this peptide compared to colistin. Colistin, they could see there was resistance to colistin, but no resistance to the peptide. They also investigated the in vitro cytotoxicity of LyeTx I in mammalian cells that is very, very low. And also it's very low the hemolytic, sorry, the hemolytic activity of this peptide is very low. And they assessed the efficacy of the peptide to induce to cure pneumonia in immunocompetent mice. And then they showed in vitro, in vivo, and in bacteria the activity of this peptide. And then they said it's a very, very good new drug. But they treated with concentrations close to the MIC, the inhibitory concentration. Sorry, estou apanhando isso aqui. And then they saw here it has a minimal inhibitory concentration of 4 micromolar. This is in most of the Acinetobacter baumannii they tested. Of course, some have a little bit more, some have a little bit less, and then this is a median or an average. And they saw it's 4 micromolar, but for the original peptide, it was 0.7 micromolar. And then the bigger peptide is a little bit better, acts a little bit better as an inhibitory concentration. But the peptide, the original peptide was much more toxic to cells and hemolytic, and had hemolytic activity. And also, they look for the minimal bactericidal concentration, that is 8 micromolar, similar to MIC 50 and MBC 50, and MBC 90 is 32 micromolar compared to MIC 98 micromolar. And then here are the colistin mix and MBCs that are much lower, because colistin is very effective, but however, colistin is very, very toxic. And then they decided to see if this peptide could act synergistically or in addition to other antimicrobials agents. And then they tested meropenem, gentamicin, levofloxacin, and colistin. And they saw that the LyeTx I Mn delta K acts synergistically with carbapenem and colistin, and addictive to gentamicin, and is indifferent with levofloxacin. And then that is interesting when you have a synergistic effect, that you can have a lower dose for treatment, because you use less of the antibiotics and less of the new drug. And they then decided that it would be a good way to use this for treatment. However, in the original work, they suggested that the mechanism of action of this peptide could be in the membrane, by lysing the membrane during the treatment, because they saw, let me see, here, using the MIC concentration, they saw a lysis of the bacterial cell, Acinetobacter baumannii, after exposure during 24 hours. And they saw the content of the cellular content in the middle using colistin and the peptide. And then they suggested the mechanism of action could be this. But they could not explain how the peptide and the antibiotics could act synergistically. Maybe there are other mechanisms of actions they proposed in the paper that could be all other mechanisms. That's why we decided to work for these other mechanisms of action. And then this was done in my laboratory by Frederico Oliveira, is a master degree, master student in bioinformatics, and Catarina Barros, a postdoc that is helping me. She is a specialist in genome analysis and transcriptome analysis of yeast, but she also works with the bacteria. And then we decided to see what kind of genes the Acinetobacter baumannii may have that render it resistant to many different antibiotics, and how the toxin or the peptide could be acting, inducing what kind of mechanisms for killing or inhibiting the growth of the bacteria. And then we use combinatory analysis using meropenem, carbapenem, and the peptide. Well, very brief explanation. In our laboratory, we use a lot of bioinformatics to sequence, assembly, and annotate genomes. And when I started this work with the Acinetobacter baumannii multi-resistant, there was no genome for this bacteria deposit in the data banks. And Maria Helena has been working with this bacteria, and I asked her, well, Maria Helena, do you have the genome sequence for this bacteria? And she said, no. Okay, let's do it then. And then it's a bacteria that was isolated from a catheter from a hospitalized patient in the intensive unit care. And it's been passing in the laboratory for many periods. I don't know how long. And then we got the DNA. We did libraries, and we sequenced using the Illumina platform. I don't know if you know Illumina platform. It's a next generation machine for sequencing and produce short reads. And then you have to assemble the short reads, and after that you get contigs for the genomes, and you assemble the contigs in bigger pieces, and after that you annotate the genes. And this bacteria has approximately 3,000 genes, between 3,000 and 4,000 genes, but interestingly, most of them are unannotated, almost 50%. And there are more than 5,000 different genomes of Acinetobacter baumannii in the data banks. And even that, almost 50% are unknown genes. That's very strange. And then we use these pipelines for genome annotation, spades and unicycle, and also you looked for mobile elements and resistant virulence genes using these tools here. We did a phylogenetic analysis on, using 121 genomes that were very well annotated and assembled in the gene bank. The bacteria we've been using is this one, AC37, multi-resistant, and we used a reference strain from ATCC called ATCC 19606, and also as an outgroup, Acinetobacter nasocomialis. And this is the what we got with this tree. And here is the, a little bit small, but the outgroup is here, the nasocomialis. This is the reference genome, Acinetobacter baumannii from ATCC, and here we have our isolate. And the red ones are the ones that are multi-resistant, and the blue ones are not resistant. And then we see that our bacteria is in a very small group here that contains multi-resistant bacteria. But we didn't know yet what kind of genes are considered resistant in this bacteria. And then we used these tools, RGI, CARD, and ARROW, and we localized 31 genes that could be related to resistance. 11 are complete genes, that means 100% similarity with known genes for resistance, that we called here perfect, and 20 are 95% identical to other genes involved in resistance. And then we saw they have mutations that could be very specific for this strain. And then we see the kinds of processes could be involved in resistance for the perfect, and especially for the perfect ones, and some strict that are 95% similarity. We localized different efflux pumps for antibiotics, and also we have other ones for replacement, reduced permeability, alteration of the target, and inactivation of the antibiotic. And also we analyzed that 121 strains here, and also the different mechanisms, efflux, inactivation, etc. The blue ones are the efflux, and the green ones are the inactivation. And our bacteria is the first one here. Every time you see black is positive for the gene, and this blue here is negative for the gene. And the bacteria we are looking for has 3 specific genes that are only seen in our bacteria compared to the other bacteria that are resistant to, multi-resistant to drugs. The ones in red here, they are multi-resistant. And then if you see here, only this bacteria, this isolate, has 3 specific genes. This was very interesting. Which genes are them? The Florex is a drug efflux exported out of chloramphenicol and present approximately in 2.8% of all Acinetobacter baumannii that are known and deposited in the gene bank. And it is responsible for resistance to chloramphenicol and florphenicol. The one AAC61B is a drug inactivation gene, is aminoglycoside acetyltransferase, and present only in 0.7% of all Acinetobacter baumannii. And it promotes resistance to amikacin and tobramycin. And the third one is ADC183, is drug inactivated, is also beta-lactamase, and resistant to cephalosporin, and is present only in 0.35% of all Acinetobacter baumannii genomes. And then that's very interesting, is a very, very rare bacteria we were working with. And we saw mobile elements, and interestingly, this gene here was transferred to the bacteria by a mobile element called integron. Probably it came from Pseudomonas aeruginosa. That's very, very rare. It was transmitted from Pseudomonas to Acinetobacter. And the other one, the floor one, it's not working. The floor one, it's not working. The floor one is transmitted by a transposon. Agora é assim. Tá. And then the floor one came from an uncultured bacteria that we have no idea which bacteria is, but it came from a transposon. And then all the 3 new genes came from transposal or integrons. And then this is the final analysis of the genome, and then we decided to look for the transcriptome from this bacteria that was treated with meropenem and also the peptide. And then we were looking for expression of the genes that are responsible for resistance. But we look at in the other genes that are involved in metabolism. And then we used a submic condition for treatment. Why we use submic? Because we didn't want to kill the bacteria. We didn't want to make problems with lysis. We want to see what kind of response the bacteria would have for the toxin. Maybe the toxin can go into the bacteria, or they can transmit the signal from the exterior to the interior of the bacteria. We had no idea. And then we decided to use submic. And we did, we tried to look for the concentrations to use with the peptide and with the peptide and meropenem. And after that, we got the RNA, we did the libraries, and we sequenced for each conditions. We had to sequence three samples that are very important. We have always to have 3 plicates from the samples. And here, we decided for the concentrations to use. This is the MIC for the peptide. And then we use MIC in a higher concentration, 8 micromolar. The MIC is 4 micromolar. And then it causes a delay in the growth. Here we saw half of the MIC, 2 micromolar, and 1/4 of the MIC. And it's very similar to the control that was no treatment, that had no treatment. And then we decided to use this concentration, 3 micromolar, because it delays the growth, but it still it's not killing the bacteria. And for the synergistic effect, we decided to treat the MIC for meropenem is 16 micrograms per ml. And then we checked 12.5, 6.5, and also with different concentrations of the toxin. And then meropenem 12.5 plus 0.5 of the toxin was very similar to the control. And then we decided to work with LyeTx I, 1 micromolar, meropenem 6.5 micromolar. That is similar to the control, but still has an action on the bacteria. And then the conditions were this condition for the synergistic, and for the peptide alone, 3 micromolar. After doing the libraries, we see how good they are. And here we see the control. The 3 libraries from control, they group together, is a PCA, a principal component analysis. This is for the cells treated with LyeTx I. And here a little bit more dispersed, but still close, we have the, this is the LyeTx I, and this is LyeTx I and meropen. And then the libraries were very, very good, more than 5 million reads for each condition. That is very good for analysis. And this is the heat map we got when we treated with the toxin and the synergy of toxin and meropenem. We got many, many genes differentially expressed. And we consider differentially expressed the ones that had at least two times fold change, is log 2 fold change 1, and also a p-value of less than 0.05. We have many upregulated genes and downregulated genes, more downregulated than upregulated in the two conditions, meropenem and toxin, and then only the toxin. Which genes are them? Here we look at the genes that are related to resistance. I don't know if you remember, we had 31 genes from the genome of this bacteria that are involved with resistance. And then we look at how was the expression of these 31 genes here. And then we see a very interesting condition. Here the 3 samples from the control, only the LyeTx I minimized, and here the synergistic effect of meropenem and the toxin. And what you see, the genes are not the same. The ones that are upregulated only with LyeTx I are not upregulated with the synergism. And also, the ones that are upregulated with the synergism are not upregulated when you have only the toxin. That's very interesting. Wow. And then, the synergism is not an addition of the effect. They have a different outcome. That's very interesting. And here are some examples of Acinetobacter efflux pump, drug efflux. And we see these genes, this ADK, AD1, I, and ADJ. They are very important for extrusion of the antibiotic, and this was upregulated in the condition for the treatment only with the peptide. And the other one is this another efflux that was upregulated when the condition was the synergism. And then different kinds of pumps are used in the synergism compared to only the peptide. And also we looked for other processes because we had many genes differentially expressed. Unfortunately, we could not see for all genes because almost 50% of them are unannotated. We have no idea what they are. And then we looked for kind of 1,500 genes that are differentially expressed, activated, or repressed. And this is the response to the peptide. What we see here, we have activated ribosome, that means translation, amino acid tRNA biosynthesis for translation, protein export, and also pyrimidine metabolism. We have no idea why pyrimidine metabolism is upregulated, but it's trying to respond the drug by activating translation. And also, we have the ones that are downregulated or repressed. And we have here many amino acids metabolism. Degradation of lysine, lysine, leucine, isoleucine, phenylalanine, beta-alanine metabolism, everything from the metabolism of amino acid is downregulated. That's very interesting. And what for? And just to show you the protein export here, the situation that is activated. There are many of the pumps and proteins involved in exocytosis in the bacteria are upregulated, the ones that are in red. And for the treatment with meropenem and the peptide, we see here downregulated. We saw enrichment in processes that are downregulated. And they are also related to amino acids metabolism, but also oxidative phosphorylation. We only saw oxidative phosphorylation downregulated when we treated with the two drugs. And that's very interesting. All the complexes of the electron chain, transporter chain, are affected and downregulated in the meropenem and the synergism. And then, I think this is the end. And then I would like to finish here telling you that you have to see in the process by molecular level. Because when you see a synergism or addiction and everything, it's not the junction of the action. They cause a different action in the end. And the Acinetobacter baumannii response, when treated with peptide, is associated with processes such as protein export and amino acid degradation, while the response to synergy is primarily related to suppression of genes involved in oxidative phosphorylation. And then we are trying to do this using proteome as well, to compare proteome and transcriptome. And this is the master project of my student. I'd like to thank Frederico and Catarina, the ones involved directly in the project, and also people from the lab that helped them. My postdoc Daniela, Jessica, PhD student, Julia, PhD student, and especially my thanks to Maria Helena that brought me to this toxin world. Very interesting. And William Gustavo Lima, the one that started all the job with the peptides, and also the help of Professor Vera Lúcia Santos from the microbiology department, and her postdoc Felipe Alex da Silva. Thank you for attention.
Thank you, Glória, for a very interesting talk. And I would like to open the session for questions.
Hope you have understood.
Very interesting results, Glória.
Thank you.
Just one curiosity. You mentioned that you found the regulation in the pyrimidine metabolism, and don't know why, but
yeah,
you
maybe you have a
you
clue.
found a lot of amino acids also regulated.
Maybe because they are linked, actually.
Yeah, you
pyrimidine
have some enzymes in common in both. So maybe it's just the software trying to please
why
you.
Why not purine? I don't know.
Because in purine metabolism, I don't think there's an overlap with amino acid
yes,
metabolism.
There are glutamic acid, aspartic acid, and glycine are responsible for the ring.
I was thinking about the valine metabolism that has overlap with pyrimidines.
Yes, but purine and pyrimidines, I don't know. But yeah, it could be a clue. Yeah. Sometimes when we work transcriptome, we have to follow the clues, and sometimes it's not easy. Yes.
Thank you.
You're welcome.
Question? Lucas?
Hi, Professor. Really nice talk. Very, very cool. Very nice work. Beautiful. I like very much. So, one of my questions was whether or not you're going to perform proteomics, but you already answered my question. So we are looking forward
actually,
to this,
we got the samples, the same samples, to do proteomics and transcriptomics, but I'm not the person to do proteomics. I like proteomics, but I don't have the expertise. And then, yeah, good. Maria Helena? No. No. And then there are...
Okay.
Yes, that would be nice to compare.
Yeah, because at transcriptome level, you have absolutely amazing data. I think it's very interesting. So, but when you look to proteomic data, almost every time you see an opposite direction of expression.
Sure.
Sometimes you have something very expressive at transcriptome level, but in proteomic level is nothing. And the opposite is the same. Is it true? Almost always. So I was wondering about this, but my question is related to regarding the proteomic field, of course. But if I get this straight, you observed that some sequestration systems are regulated in your transcriptome level.
Yes.
I work with bacteria in my master's, so I studied a lot of sequestration system. I think it's sequestration system type 2 that secrete a lot of toxins and bunch of things. So you wonder, together with this proteomic data, you could consider, of course, to study the exoproteome, what is secreted from this bacteria.
Yeah, that was
because
one.
It's very interesting because there are a lot of things that could be regulated.
Actually, we would like to do the exoproteome, the secretome. I don't know if you can do that, say that, secretome. But also, yes, secretome, and also the metabolome, to see how the metabolic products will be changing by the use of the peptide and the mixture of and meropenem. But actually, I don't have expertise in protein, neither the metabolomics. It's very hard for me. I have colleagues in the institute, but my laboratory is focused on DNA and RNA and not only in proteins. But that would be amazing.
Yeah, but it's very nice. I really appreciate your work. It's very, very beautiful. Congratulations.
I'd like to comment a little bit about, you said, the correlation between proteome and transcriptome. This is worse for animals, for eukaryotes, because for eukaryotes, the RNA is produced in the nucleus, it suffers processing, and then transported to the cytoplasm, and the protein synthesis will be there. And then there is a lag period between the synthesis and the translation. And the RNA has half time, different from the proteins. That's why when you look at the same period, same time, you don't have to see the correlation. Sometimes the RNA is going to be translated in the future. And the other thing, I work with eukaryotes and gene expression regulated post-transcriptionally and co-transcriptionally by splicing. Sometimes you produce isoforms from alternative splice that are never translated.
It's a nightmare.
Yes.
I understand. I work with eukaryotes.
Yeah, but for bacteria, could be more direct, because the bacteria transcribes and translates immediately, and then maybe you can see a more positive correlation. Yes. Don't know. We have to see that.
But it's very, very interesting because you're trying to do what is called today multiomics perspective. Because it's trying to integrate everything. I wish all the luck you could bear in your life.
Thank you.
Maria
Helena.
Thank you, Glória, for the very nice presentation. But I would ask you an intriguing question. With your results, do you think that this peptide is cross the membrane, surely or not? Could you affirm this?
Yes, I think so, because we have stimulated pumps that go do the efflux, like the systems to transport the drug to the outside, and then the drug enters.
So you
Well,
cannot
we
give,
have to prove it enters, but stimulation of different kinds of pumps, not only the meropenem and the peptide, but the peptide alone, it's an indicative that
not
it
possible
enters
to be only
and the
by
bacteria is trying to extrude.
not only by the membrane.
No, I don't know. Probably, I don't know how could
Yes,
it
my
be
question is this.
coming into the cell by the membrane. I don't know if it's by endocytosis, how can I say that? Endocytosis or transpassing the membrane.
Crossing the membrane.
Yeah, could
Because
be.
it's antipatic peptide.
Yes, actually,
antipatic
it has
peptide.
positive residues
Yes.
and
the hydrogen, the neutral.
not pollen. Transient pores could be, but it's good to label the toxin and see how it works in the membrane. Yeah, but I think it goes inside, and the bacteria tries to send it to the outside.
Questions? Okay.
If the students want to ask in Portuguese, you can ask in Portuguese. No, not you. I mean the other students. No, but you I know.
Eu falo pouco português.
No, you can ask in English.
Okay. Okay. Once again, it was a wonderful presentation, very comprehensive, especially in microbiological studies. I just want to see your reaction on the synergism assay, as you tested the peptide with antibiotic, right? So it's just a curiosity, because I'm using the same peptide, but I will conjugate it with the polymer. So what I can expect, either I expect synergism assay, or maybe it reduces the antimicrobial efficacy, or it is the same as it is. So what I can expect.
Yeah, I think you have to check. Yeah, because when William did that paper in 2021, he checked with different antibiotics and saw some that they have synergism, other ones addictive, and other ones are indifferent. No one was the opposite, but you have to check. Yeah, it's the only way to see.
Yeah. Thank
And
you once again.
and you are lucky. William is there with you.
Yeah,
You can ask his
no
help.
Doubt, no doubt it is.
William is a very, very nice microbiologist.
Wonderful guy, yes.
Yeah.
It is. Thank you so much.
You're welcome.
Thank you,
Thank
Glória,
you.
for an amazing talk, and congratulations for the work. And have a nice trip back home.